Timer పెట్టుకుందాం. వద్దు జస్ట్ టైమర్ స్టాప్ వాచ్. Okay, start. Good morning. We are the students of BSc Biotechnology, Indira Priyadarshini Government Degree College, presenting you the Jignasa student project. Title: Immunoinformatics Guided Mapping of Epitopes in Target Proteins from Trichomonas fetus. The project includes introduction, material methods, results discussion, summary, and conclusions. India has the largest livestock population in the world with 528 millions of domesticated animals, but the conception rate is only 35%. Potential risk of sexually transmitted diseases and the impact on conception and animal breeding is paid very less attention. Most of the diseases are zoonotic, pose public health issues. The burden is expected to be more than $1 billion. As per the recommendations of Department of Animal Husbandry, Fisheries and Dairying, a list of diseases have been made mandate to screen in bovine stations. Among the diseases, bovine trichomonosis is neglected and paid very less attention. The geographical distribution of bovine trichomonosis seems to be mostly worldwide. Most of the studies have been published from North American, United Nations, Canada, Argentina, Australia, etc. Limited studies have been conducted in India due to lack of point-of-care diagnostic kits. Only the commercially available diagnostic kits include bovine trichomonosis test kit, in-pouch TF culture kit, and VMAX gold rich detection kit. Since these kits being very costly, they cannot be affordable at the point of care testing. Hence, there is a need to develop most viable point of care diagnostic kits like ELISA and rapid kits. If we look into the physiological structure, pathophysiology of Trichomonas fetus, it is a parasite with 3 anterior flagella and 1 posterior flagella with an undulating membrane nucleus. Belongs to the kingdom Protista, and it has genus Trichomonas, and the fetus is the species. If you look into the transmission of this parasite, it gets transmitted within the herd from cow to bull as well as bull to cow during the natural mode of mating. So if one animal is infected in the herd, it is like almost 30 to 90% of the animals get infected. Clinical symptoms include the signs of early abortion, high percentage of unbred cows, repeated requirements of mating for conceiving, irregularity in estrus, mucopurulent vaginal secretions are the most prominent symptoms, and they account for 20 to 40% of reduction of the income source from the cattle breeding. There were several reports of zoonotic transmission were reported in human, leading to different diseases associated with them like pneumocystis pneumonia, meningoencephalitis, peritonitis, and cholecystitis. In view of the above circumstances, we have laid the following objectives: to retrieve the amino acid sequence of test proteins from NCBI, to perform the functional annotation of selected proteins, to predict the subcellular localization of test proteins, analyze physicochemical properties, and to analyze the selected proteins for epitope domain prediction. The outline methodology include retrieval through NCBI, and let us get this. So we have shortlisted 10 most suitable proteins based on the earlier publication by Curley et al. 2020, and we have performed the functional annotation using UniProt KB, and we found all the 10 test proteins found to be highly involved in antigenicity, virulence, and establishing host pathogen interaction. Further, we have done the subcellular localization to predict the localization of target proteins within the cell, and we found that most of the proteins are found to be present in the cytoplasm, present in the plasma membrane, and few proteins were found to be secreted extracellularly. And we have performed membrane localization prediction using TMMHM tool, and we found that cysteine protease 8 is an extracellular protein with, and several remaining all other proteins were found to be transmembrane protein with one or more transmembrane domains. We also found circumsporozoid protein, immunodominant variable protein, to be found to be membrane associated proteins. Further, we have done prediction of physicochemical properties using XPASI protparam by submitting the FASTA sequence, and we could found the molecular weight, theoretical PI, and stability of the proteins when expressed in yeast. We found all the target proteins found to be stable for more than 20 hours when they are expressed in the yeast cells. Finally, we have predicted the epitope domains of target proteins using IEDB tools, BiViPRIT linear epitope prediction 2.0. And we have noted the epitope domains that were graphically represented at the threshold more than 0.5 for all the proteins. And finally, we conclude, and this is the table that shows predicted domains of different epitope domains present in a particular protein. With this analysis, we conclude that cysteine protease is the most suitable antigenic protein due to extracellular secretion with several antigenic domains. Multiple types of cysteine proteases were involved in pathophysiology. T. fetus parasites tend to inhabit the genital foldings in the early phases of infection, secrete proteases into genitals for tissue invasion. Transcriptomics proteomics studies have also reported high fold expression of cysteine proteases. And among the cysteine proteases, we found after cysteine protease 8, CP13 and 16 were found to be highly expressed among all 20 cysteine proteases. Thank you.
6 7 minutes approximately 7 minutes.
మీరు ఇంత చెప్పలేరు. కాకపోతే ఏం తగ్గించడానికి మీకు అర్థమవుతుంది కదా, మనం ఏది మాట్లాడినా 10 ప్రోటీన్స్ గురించి మాట్లాడాలి. సో లెట్ మీ స్టాప్ రికార్డింగ్. వేర్ ఇస్ దట్
ఇంట్రొడక్షన్ ఒకటి, నెక్స్ట్ రేణుకా 11 to 16 సైట్స్ కొంచెం ఎక్కువ.
ఏమ్మా వేర్ ఇస్ రికార్డింగ్ పాజ్ అయిందా? అసలు రికార్డింగ్ అయిందా దీంట్లో? షేర్ పాజ్ రికార్డింగ్.